5 years as part of what was President Mary Sue Coleman's clusters hires approach. The idea was to have 3 or more departments or schools of the university come together, identify an area that we thought was really significant and likely to grow in the near future in scientific importance, and have a sort of simultaneous recruitment for those different departments and stimulate as much interconnection as we can among the faculty once they're here. So I've taken on this mission with the biological networks cluster. A month ago or so, many of you heard Megan Duffy and Deepa Rajapakse talk about their research in this domain. And today we're going to hear from Kevin Ward and Kevin Wood. There's another guy here, Kevin Ward. The way it came out to my ear. Yeah, right. I hope I said it right. And Marisa Eisenberg. They will be introduced by your colleagues from our student body who have taken charge of this whole process. And I don't want to steal any thunder from them. So let me turn it over to you.
Thanks, Gil, for the introduction. And I would like to just say a few words about our first speaker, Dr. Marisa Eisenberg. So she did her PhD in biomedical engineering and something which sounds very cool, cybernetics, in UCLA, and followed by a postdoc at the Math Institute in Ohio State. And she focuses on like multi-scale modeling of like disease propagation and their viability. And while looking at some of your papers not too long ago, something which caught my eye was something called X-flu, which I thought was very cool. So I think this is very generalized view, but they kind of use cell phones and Bluetooth technology to find out how students are connecting with each other and how that relates to like subnetworking and spreading of influenza-like diseases. So I thought that was very cool.
Awesome.
Yeah, and I think this is very cool that you're involving all the multi-scale networking, like from cells to population and all the different temporal timescales. And today we'll be hearing about identifiability and parameter estimation in modeling disease dynamics.
Awesome. Thank you. Thank you for that awesome introduction. That was fantastic. So yeah, I sadly wasn't planning to talk about the X-flu stuff today, but we can talk about it in the panel discussion. It was exactly as you said, we're using sensors and cell phones to track people's contact patterns and track things like their hand hygiene behaviors and things like that, so that way, you know, we can say like, oh, Marisa, like if I'm coughing, like we can have sensors that might detect coughing, and then we can see that you and I were hanging out, and then in a couple of days, you know, we can ask and say, hey, so how you feeling? You know, like if you were interacting with someone who might be sick and those kinds of things. So tracking people's social networks and then looking at how diseases spread over those different social networks. So that's one area that we're working on. But today I was going to talk sort of more on the mathematical modeling side about a problem that I'm interested in on identifiability. And I thought I would start with a little, this is like a very silly kind of baby basic, you know, thing about why we build models. And all of you I'm sure know, but sort of one of the like foundational purposes of models is in some sense to act as like a conduit between our theory and our mechanisms, how we think things work, and the data that we have, right? So that's sort of one of our major purposes. And so then we use models then to try and understand the details of the mechanisms, things like transmission routes, causality, and also to try and sort of help maybe guide or test treatment alternatives, intervention strategies, different things like that, and then as well for forecasting and prediction. And so, so some of the things that we use modeling for, but in this process, since we are connecting with data, there are inherent issues of uncertainty, right? So it's not actually always clear that you can actually estimate or get all the information that you want from a given model and data, right? And so we're often not actually measuring the thing that we're interested in. That's part of why we're building the model, right? Is to understand the relationships between the variables that we maybe aren't necessarily measuring. And so how much can we actually infer about those things, and what data sets do we need? So those kinds of questions are the kinds of questions that I'm excited about. And that's part of why, gathering network data, different kinds of things like this, how do we use models to understand how things work? How do we combine them with different kinds of data sets? And what data do we need to actually get the information we're interested in? So those are the kinds of questions that I am excited about. And so I thought I would mention today sort of a little bit more about this identifiability question. So I usually ask people, so how many people are familiar with identifiability as a thing? A few of you, okay, but not all. And so yeah, so identifiability is addressing this question of whether it's possible to uniquely estimate your model parameters given a model and a data set, right? And so framed mathematically, we can sort of think about this as injectivity of a map, right? So you can imagine your model as really sort of being like a map from your parameter space to your output space or your data space, and we want to know if that map's injective, because if it is, then we can get from a data set, we can pull back to what parameters gave it to us. And so identifiability then is asking this question, and there's a couple of different kinds of identifiability that people often talk about. So practical and structural. So I usually sort of walk people through this a little bit. So practical identifiability is all the things that can just due to like quality of the data, maybe frequency of measurements, number of replicates, all these kinds of things, all the reasons that data that maybe could be informative maybe isn't as informative as you would like for practical reasons, right? But there's also a thing called structural identifiability, which has a lot of interesting properties. And so this is getting at, is the structure of the measurements that you've chosen and the model you've designed, sort of not well posed to be able to actually estimate your parameters, right? And so the like silly example that I use in all my talks for this is this simple linear example. So imagine you have these red, I have a pointer, I can actually do this. You have these red dots are your data, right? And then the model you propose is y equals m1 plus m2 times x plus b, right? Then no matter how perfect this data is, you can never estimate m1 and m2 simultaneously, because any value of m1 and of m2 that gives you the same overall slope is going to give you the same fit to the data, and so you can't estimate these two parameters independently, right? So, so we would say these parameters are unidentifiable, but of course you can measure their sum, right? So you can estimate the sum of m1 and m2 because you can get the slope, but you can't estimate the individual parameters. So oftentimes with structural unidentifiability, what's really happening is that there are dependencies between your parameters in terms of what they're affecting or how they operate. And so then you may not be able to estimate individual parameters, but you can estimate combinations of them. And so that's a lot of what I work on. Now, in obviously this case was sort of silly, and in any sort of real models that we would really be considering, you're not looking at things where they're obviously grouped in one term together. These kinds of combinations can show up in different equations. They may not go away when you non-dimensionalize. There's a lot of different things that can go on. And so, so this is something that comes up a lot in a lot of different contexts when you're working with mathematical models. The structural question is sort of a best case scenario if you have perfect data. That's obviously unrealistic, but it's also a necessary condition for being able to actually do parameter estimation with real data. And so there's a ton of different approaches that we work on and that other folks work on for trying to figure these kinds of questions out. And so they can run from the more theoretical. So I'm not going to actually dig into most of this. I just sort of put this here almost as a placeholder. Some of the things that we do are trying to figure out the sort of shape of these combinations, what these combinations might be. And so a lot of this actually relies on tools that are pulled from computational algebra. And so you can use you can, if you're thinking of like ODE models or certain kinds of stochastic models, you can actually view your system in a polynomial ring and then do a bunch of computational algebra to figure out the injectivity properties of your map. So there's a bunch of sort of neat approaches like that that let you take advantage of what were sort of traditionally more pure math tools that we can use here. But there's also a bunch of analytical, or pardon me, numerical approaches that you can also use. So things like the Fisher information matrix and that kind of stuff, if you're familiar with those concepts from statistics, stuff with the profile likelihood, ways of essentially looking, if you're doing fitting, right, then you have some surface of, sum of squares or a likelihood surface or whatever it may be, and by looking at the shapes of that surface, you can answer a lot of these questions that way too. So that's the kind of stuff that I work on. Oh, wait, wrong one. There we go. And so I thought I would just, since this is meant to be sort of an introductory talk, I was taking this as it's meant for a discussion, right? So like I wanted to just pose some questions that I like, and then maybe hopefully they intersect with things that you are interested in, and we can discuss some of those things, and then I'll talk about a couple of applications. And so, hang on, how are we doing on time? Okay, so some questions then that I often am thinking about, whether I'm doing modeling or some of these more like social network types of applications, different things, are what information can we get from a given model and data? You know, what is the structure of the dependencies between parameters that we're trying to estimate? And how can we use these parameter dependencies to better understand our system, decide what data we should be taking? So maybe you really care about this parameter because it tells you what dose of drug you should be giving. If that parameter is unidentifiable from the setup that you have, you may want to change what you're measuring, right? Like, and so knowing that ahead of time can help you decide what you should measure. And so we can use these kinds of approaches to think about that. And sort of more towards that kind of practical question, then there's also these issues of how does your parameter uncertainty influence your ability to make forecasts or predictions, your ability to decide on a control strategy or an intervention, or what inferences you're going to make, right? So, you know, for instance, if you imagine, let's say like with that M1, M2 case, right? Every value of M1 and M2 that gives the same total sum, right, is going to give the same fit to the data, right? And so you can have different values of these parameters, and maybe the control strategy that you would want to do, like the intervention or treatment that you'd want to do, is different for those different parameters, right? In which case, maybe we need to resolve that unidentifiability, right? But maybe it's not. Maybe you can use the same treatment strategy across all of that space, and as long as it stays fixed over sort of this curve of parameters that all give the same fit to the data, then we don't care if the parameters are super uncertain and we can't know what they are, right? We know what to do, so who cares if the parameters are unidentifiable, right? And so, you know, questions like that I think are very interesting, and you'll often see cases where it's not always clear and you have to kind of work through it about whether actually the quantities of interest or the prediction or the control strategy are conserved over this space of parameters that fit the data well, or they change, and then what do you do if they change, right? So maybe instead of doing the optimal control, you pick some sort of bet hedging strategy that lets you not do as well as you could, but it works better across the range of all the possible parameter values you could have, right? So things like this I find very interesting. And so those are the kinds of questions I like. And so that's sort of the intro to identifiability as a thing. And then I thought I would talk for a few minutes just a little bit about cholera. I could actually switch. I think, so I should be wrapping up by what, like 4? Is that about when I should wrap up-ish? Or, okay, yeah. But so I probably don't have time to do both. I have 2 applications. I have cholera and Ebola. And we could do either one. It's an odd question, maybe. But so I guess maybe we'll start with this and we'll see. But we could. Do people have a preference? Well, so we think. Yeah. But yeah. Okay. So maybe we'll talk a little bit about cholera. And maybe that's of interest anyway, given like Hurricane Matthew and all the recent stuff. So okay. So let's talk about cholera. So cholera, just as a little bit of background about the disease, it's a waterborne intestinal infection. It causes 3 to 5 million cases a year, over 100,000 deaths every year. You probably saw, you know, the large outbreak in Haiti in 2010, right? So, but so people often sort of think of cholera and they imagine like Jon Snow and the Broad Street Pump in like 1800s London, but it's still very much a disease of today. And it's a waterborne disease, as I mentioned. It causes this profuse watery diarrhea that leads to dehydration and can be fatal quite quickly. But it's easy to treat. You just keep the person hydrated, either orally or by IV. The issue is usually that when you have poor infrastructure, it can be hard to get the person to somewhere where they can stay hydrated well with clean water, you know, rapidly. And so one of the things that is of interest for us when it comes to thinking about cholera is the fact that it has these multiple transmission pathways. So it's a waterborne disease, so a lot of the transmission is happening through environmental water sources, but it can also be transmitted through foodborne pathways, household water sources, all sorts of other different pathways, right? And so for the purposes of the model that I'm going to talk about right now, we're going to sort of clump the transmission pathways into sort of two categories: environmental, which uses these, outside environmental water sources, and everything else that's sort of more direct. So foodborne, household water, all of those kinds of things. And so the really simple model that we're going to talk about right now is this SIWR model. So how many people are familiar with SIR models? Some, but not all. Okay, so SIR models are these classic epidemiological models. They're differential equation models. They started in 1927 with Kermack and McKendrick's very first SIR model back in the day. And they've sort of expanded and become a very commonly used tool in infectious disease epidemiology to study disease dynamics and outbreaks and things like that. So the way that these models work is that you typically, you clump people into groups based on their disease status. So here we have susceptible, there as S's, infectious people who are I, and then recovered people who are R, okay? And so you can progress from susceptible to infectious to recovered, and that's the classic sort of SIR model. The difference here is that we've added an additional compartment W, which represents the concentration of bacteria in the water. And so now, instead of just transmitting directly when an S and an I interact, you can also transmit by having an S, a susceptible person, interact with the bacteria in the water and get sick that way. Once they're sick, they can shed into the environmental water source and then, transmit further. Yes, good. Oh, that's a good question. That would actually be quite interesting to put like a, Oh, yes, good point. Oh, into the mic, I think. So yeah, are there cases where the W is actually mediating the transition from I to R, thinking of W not as pathogens, but as water and the availability of clean water? That is actually a great question. I don't know that people have modeled, like this is a thing actually, and I'm not going to go on this soapbox, but in epidemiological modeling, one thing that we often neglect is thinking about the logistics. And so how does the availability of clean water affect the dynamics of your disease and things like that. And so we haven't modeled that particular thing, but you definitely could and maybe should. Yeah, exactly. Yeah. I mean, we model interventions often that way, but you don't often sort of, So, yeah. Anyway. Okay, so this is our simple model. You can write down a system of differential equations for it. You can calculate things like, if you're familiar with stuff like the basic reproduction number, these sort of key epidemiological quantities that you can calculate. And so if you look at the identifiability of this model, the model is actually structurally identifiable. So meaning that with perfect data, if you could measure, say, the prevalence or incidence of disease, then you can estimate all the parameters in theory with perfect data, but actually you can lose that identifiability under a couple of different circumstances. So one case is where if your decay rate of your pathogen gets too fast, then the timescales for waterborne and direct transmission can become too similar, and you stop being able to distinguish which transmission pathway is actually which. And so this can actually have implications for how you decide what interventions you should do, right? Because you would use different interventions if you think that a pathogen is primarily being transmitted person to person versus if you think it's primarily being transmitted via the water, right? And so you can lose that identifiability. So here, the color here indicates the goodness of fit, with blue being better, a better fit. And so you can see that for slow pathogen decay rates, you have sort of one single minimum, and you can distinguish the transmission parameter for the water from the transmission parameter for person to person. But once that decay rate gets too fast, you lose that identifiability, and you can only estimate the sum, the total amount of transmission that's happening, right? So things like that can be an issue. In the case of cholera, the decay rate is quite slow. So we're much more in this regime than in this regime. So you would think that your identifiability should be okay. It turns out that actually there's still additional practical identifiability problems that can crop up. And so you can have issues where the transmission parameter from the water and the decay rate of the pathogen in the water can actually trade off from one another. And so these black circles here indicate various runs of parameter estimation with the true value shown in as a red star here. And so you can get these sort of trade-offs, even though most parameters are estimated well, certain parameters are not. And so then it actually can affect your ability to, again, measure things like the contribution of different pathways, your ability to measure classic epidemiological quantities like R0, all of these different kinds of things. And so you can also then look at what information, what data should you add to improve your identifiability and fix your parameter estimation. And it turns out that if you measure the concentration of pathogen in the water, so you're not just measuring how many people are getting infected, you're also measuring the concentration of pathogen in the water, then now you can actually estimate your parameters just fine, and it improves your identifiability. And we've started actually doing this in a case with polio, where we actually have environmental surveillance data, and you can really do this properly with time course measurements of both pathogen and people. But how are we doing on time? I thought I would talk a little. I've got 5 minutes. Okay, so I can do this in 5 minutes. So I thought I would talk a little bit on the applied side. I guess actually, what are people interested in? Are people interested in the applied side of things? Yeah. Okay. Good. All right. So I never know how theoretical versus how applied to pitch things. So when it's a blended audience, it's good, but it's also confusing. Okay. So Haiti, there was a cholera epidemic that started in October of 2010. Haiti had basically sort of not seen cholera for an extremely long time prior to this, possibly ever. It really sort of took the whole country by storm, and it sort of spread across the entire country and has actually stuck around since. And so there has been a series of epidemics each season, every year since. And so we started working on this sort of shortly after the epidemic started. And these are just a couple of photos from the Artibonite Valley, which is where the outbreak began, and from a cholera treatment center, some of the displaced person camps, so the tent camps around Port-au-Prince and different things. And so we were interested particularly, so one of the things that people often ask for when you are modeling an ongoing epidemic is to try and get a sense of the basic reproduction number, this R0 kind of quantity. But the other piece is often forecasting. So that's a common thing that people are interested in. And so we were interested in using environmental data, knowing that adding water information about the pathogen in the environment, things like that, could improve your identifiability. We were interested in adding environmental data into our models to see if that would help us with forecasting. And so you can see, cholera is well known to have a lot of environmental drivers. This has been something that a lot of the folks here at Michigan have worked on for a long time. But you can see it here for this epidemic. You have cases in Haiti, going along, and then Hurricane Tomas happens, and you see this sudden upswing in the number of cases that there were, right? And so we added into that very simple model that I showed you spline fit precipitation data from the NASA Tropical Rainfall Measuring Mission. And so we sort of just added that in the silliest way that you could, the simplest way, just putting it into this waterborne transmission term, sort of with the thought being that when it rains, people are more likely to come into contact with infected water sources. Those water sources, there's fecal matter and stuff that gets flushed into those water sources, and so you would expect to see an upswing in the amount of waterborne transmission. And so, so we included that into this model, and then, so here the black line and the gray data are the data and the model that we use for fitting. The model forecast here, these are just a few examples, is shown in red, and then this is what actually happened with these red dots. And so you can see that you can actually do a pretty decent job of forecasting how many cases you expect to see if you have information about how much rain you're going to expect to see as well, right? And so these are in 4 different locations: in Hospital Albert Schweitzer, in the displaced person camps around Port-au-Prince as a whole, and then nationally. And so, so you can do some stuff with this, and it actually sort of works. And I think maybe with that, yeah, I'm going to actually wrap it up. I was going to tell you, we also did some, so I was also going to tell you a little bit about a vaccine campaign that we helped to design in a refugee camp on the Thai-Myanmar border. So there's this Mela refugee camp, which is the largest refugee camp in Thailand, and it's mostly Karen refugees. They were having cholera epidemics every 1 to 2 years, and so we helped with designing a vaccine campaign, figuring out what dosing strategy. The cholera vaccine requires 2 doses, and it's a bit awkward, right, because you don't necessarily have a good count of who's where and who's who. And so there's this question of do you need to actually follow each person? Like, hi, I gave you a dose, and now I'm going to come back in 2 weeks and find you and make sure I give you a dose? Or could we do 2 waves of just first come, first serve and make sure that we actually caught enough people? And so we were able to sort of help with the vaccine campaign design and use some of these same identifiability methods to figure things out. I'm going to skip right through all of that and basically say they ran the vaccine campaign. They actually did not have an epidemic the following summer, which was great. We ran a second set of simulations the following summer after that to see if they would need to run a booster campaign, and we're able to sort of recommend that, like, no, it didn't look like you needed to. They didn't have an epidemic that summer either, so it was a nice sort of project to work on together. But I'm going to wrap it up instead and not talk about Ebola and skip instead to my thank you slide. So I guess just to finally sort of wrap things up, I think there's a lot of interesting uses of modeling and connecting models with data. I'm also very excited about new kinds of data, whether that means, you know, satellite data or that means social networks and, you know, phone sensor kinds of data, things like that. I am excited about thinking about how models can connect with those kinds of data sets and what information you can get from that. And with that, I'll just thank my collaborators, my students, the folks who provided us with data, and also our funding. And yeah, that's it. Thank you.
I was just reading something very naive. So I was just reading a paper from James Setna's group at Cornell, and they talk about and so it's a lot of the same ideas where they're looking at models at different length scales, and they connect it back to this idea in statistical physics of renormalization flows based on. So I was wondering, do you use models that are multiple? So the idea is that you don't have to know every parameter necessarily to describe a system, and it's some hope of why simple models can work. I guess my question is, do you look at models over length scales, whatever that means, to combine parameters and look at things like that? Do you directly and the information geometry and stuff like that?
Exactly. So they're asking a lot of their questions are similar to. Oh yeah, sorry. I should be good at that thing. A lot of the questions that they ask are similar. So do you guys know about this stuff? So there's this group, James Setna, and they do a lot of stuff with they call it sloppiness, which like can mean that there are certain directions that parameters can sort of compensate for one another in, and you can get, it's a very similar, they have a slightly different metric, but it's a very similar idea to this sort of unidentifiability.
Okay, that's what I was wondering.
And so, yeah, and so you can do, a lot of their work is about trying to like reduce your parameter space and reduce your model, and oftentimes that ties into the idea of different scales, because it's very common for, based on scale, for you to be able to sort of do these kinds of model reduction. And we do a lot of that more with HPV. So in those cases, we're thinking about, there's actually a lot of, I think, sort of interesting questions when it comes to multi-scale stuff with thinking about infectious-driven cancers. So because you have population scale processes happening with the transmission, right? But then within host cellular level processes happening with the carcinogenesis, right? And those are also happening on wildly different timescales. So you can have the infection process happens while you're young, and then it takes 20 years before you actually develop the cancer, right? And so there's a lot of interesting things there, and you can see like fun collapsing of parameters so that as you look in one time scale or one length scale or another, different simplified models are what you need. Yeah. So,
cool.
Yeah.
We should talk more about that.
I know.
So thanks for the great talk. And our next speaker is Dr. Kevin Wood. Dr. Wood did his PhD in theoretical physics and chemical physics at the University of California, San Diego, right? And did also his postdoc in Harvard about molecular and cell biology. And he has a strong background in both quantitative and biological sides. And he had a great experience with your younger brother on bacteria drug resistance analysis, right?
That's right. That's right. Yeah.
Very interesting. And also your research involves in game theory application on bacteria population. And today he's going to share his idea about population density and bacterial drug resistance. Thanks.
Thank you. Well, thank you for the invitation and the nice introduction. So I've met several of you before. I see some familiar faces. I've been here about 3 years, and it's really easy for me to keep track of because my son was born right when we were right when we got here. And so like about a week and a half ago, he had his 3rd birthday. So I have a living and breathing tenure clock that is growing very quickly. But it helps me keep track of things. So the types of questions we ask in my lab are how do population dynamics contribute to things like antibiotic resistance? That's just one particular problem. And by population dynamics, I don't mean populations of people, I mean populations of bacteria. So a common approach to studying a problem like this, and this approach is absolutely essential, is to zoom in if I have a population of cells, and then keep zooming in and saying what's going on inside of the cell that's causing the drug to work and maybe causing the drug not to work under some conditions. So the goal here is to understand molecular mechanisms. One example is like a cell may make an enzyme that degrades a particular class of antibiotics. There's an enzyme called beta-lactamase that does that for penicillin-like drugs. What you can do then is this enzyme would render the drug ineffective. Now, the way we like to think about it is that this knowledge is absolutely needed, it's absolutely essential, but maybe it's incomplete because bacteria don't live as isolated cells. So let's say I go back to my population and I say only 2 of those guys make the enzyme. So these would be 2 resistant cells. In some sense, these cells can be altruistic in that they can detoxify the local environment. They can get rid of the drug and potentially help the other cells. So then we've got this situation where we have sort of a bimodal distribution of phenotypes where some cells are resistant, but they're sort of paving the way for the other cells to survive too. So it sets up this sort of cheaters and defectors dynamic. There's a beautiful paper from Jeff Gore's lab at MIT a few years ago describing this in well-studied mixtures. The idea that I want you to take home is just that population behavior can be really rich, and that our goal is then to take these molecular mechanisms, often that are known, sometimes not, and try to understand and extract from those what happens at the larger population scale in bacterial populations. Now we use a lot of times for that mathematical models. Many of these are based on statistical physics. This was a theoretical training that I had as a graduate student. Statistical physics is, people often think of molecules and atoms and things like superconductivity, which is really cool stuff, but it applies more generally to interacting systems. So some examples where statistical physics approaches have been used are birds flocking. So you see these like beautiful patterns. If I want to understand birds flocking, yes, I need to understand how individual birds fly to some extent, but to really step back and understand this, I need to know more than just how a single bird flies. I need to know how it interacts with the other birds, right, to form the pattern. Ants can do this. This is a movie I found online. This isn't the whole movie, but when the phone rings, the ants start moving, and when it stops ringing, they scatter. So I don't know what this means, but even the Supreme Court, there was a paper called The Statistical Mechanics of the Supreme Court about a year ago from Bill Bialik's group at Princeton. The idea is if you know the political leanings of each justice, you actually can't predict very well the consensus that the court is going to come to. This was taken over some 10 or 15-year period. But if you account for interactions between justices, say Justice 1 and 2 tend to vote together or they tend to vote in opposition, then you can suddenly predict the results really well. As a postdoc, I did something similar, actually used the exact same mathematical approach to show that something similar happens in bacterial populations when you apply drugs. If you know the effects of drugs individually, it's very hard to predict what happens when you put the drugs together. You may have a drug that works great, another drug that works great. You put the drugs together and often the combination won't work anymore. It's called an antagonistic effect, or the opposite case happens. So while it's very hard to predict at that level, if you look at larger combinations of drugs, what you find is that pairwise combinations are often sufficient to predict that. So it's the same type of idea here. Again, sort of what I want you to take home from this is that there's a lot of powerful mathematical machinery out there for dealing with interacting systems. This is probably my favorite statistical physics book. It's not necessarily the one to start with, but it's a really beautiful one. So in addition to these mathematical tools, we have to be able to make quantitative measurements. And bacteria are great because you can make a lot of quantitative measurements with bacteria, right? We don't have to go, we don't have to go abroad. We don't have to try to track people. We have in very simple setups a way to make very quantitative measurements. What we've done in the lab is we've built basically an extension of what was developed 60 years ago or more, something called a chemostat. A chemostat just means you keep the environment constant. This is an example of our chemostat. It's a little more complicated. This was built by a graduate student, Jason, in my lab. This is really, really nice work. It extends over more of the lab, but it just looks messier and messier as we put it all together. The idea here, if you look more closely, is that you have here a little cup holder, essentially. We got these done at the physics machine shop. And you have a glass vial that goes into the cup holder, and that's just me holding the vial. And at the top, you have tubes. We connect those tubes to peristaltic pumps that can pump liquid in and out. You connect those pumps to a series of computer-controlled data acquisition cards, and now we can control environments very precisely. We can pump in drug, we can change pH, we can change media, and things like that. We can also measure, at least roughly, how many cells are in there. We do that by drilling a hole in the side of these. We put a little light and a detector pair. The first version of this actually came from Radio Shack. It cost $1. We did it, and we compared it to our $40,000 spectrometer that takes optical density readings, and the correlation was almost perfect, right, over a wide range of density. So this is basically a way of measuring optical density, which is proportional in many cases to the density of cells in a population. So we can track that, and then we can simulate more complicated situations by connecting these vials together. You have these vials connected together, and you can simulate different flow rates, migration. You can start to study things about how evolution happens, evolution of beneficial traits in general, the effects of spatial heterogeneity, things like this. Okay? Any questions so far? So the first thing we did with this setup is we said, okay, well, let's start with something that should be really simple to measure. We said if the response of drug really involves some types of cell interactions, then we can probably guess that the overall behavior, and by behavior I mean the response to say, antibiotics, which is something that we were interested in, should depend on population density, because that sets the length scale and possibly the magnitude of these interactions. Shouldn't that happen? And of course, if you go to the literature, you find that yes, it does happen. This is called the inoculum effect. It was discovered something like 50 or 60 years ago. It's been known for decades. What the inoculum effect says is it says if I start with a higher density of cells, a higher inoculum in my culture, and I measure the minimum inhibitory concentration of drug, the amount of drug needed to essentially kill the population, then I find that MIC goes up with population density of the initial starting population. Now the problem is this is a little bit difficult to interpret, because if you think about these experiments more carefully, you realize that density is changing by many orders of magnitude on the time scale of these experiments. So it's very tough to say, what is, say, population growth rate as a function of density and drug concentration? Right? So as a result of this, this is something that's often cataloged in the description of new drugs, but rarely used in any clinical situation. It has been shown in some animal models, so it does seem to have some effect, but it's rarely used. Part of that is that this functional relationship is missing. With this type of setup, this is something that's very easy to measure. So this is work from Jason and Jeff, 2 graduate students and an undergrad in my lab, Pete. So what you can do, we use a particular bacterial system that's called Efficacy or Efficacy. I'm not going to focus on that too much here. But what we can do is we can hold the density of a cell population constant. So this is a growth curve. This is optical density. It's proportional to the number of cells loosely. This is time. The red is just a typical growth curve, just an exponential growth curve. Or we can say we want to hold this population at a particular density. So while it's growing up here, all of a sudden, we use feedback control to pump in new media and pump out the cells. Okay? And we can keep track of how often those pumps or how hard those pumps have to work to keep this density constant. Based on how hard those pumps have to work, we can then infer the per capita growth rate of the population. We can now do this as a function of cell density. So that's exactly what we set out to do. We said, we'll apply a drug and we'll see how does that drug act at different cell densities. And when you do that, you see there are huge effects for many drugs. So this is the growth rate over time. We add drug at time 0. You have some transient effects where the drug has some effect that we're not exactly sure what's going on, but then it settles into some steady state growth rate. The growth rate changes as the optical density or the population density of cells changes. So we can take something like this and we can plot it. This is just growth relative to untreated cells as a function of cell density. And the fact that this curve goes up or down in some cases tells you that there's an effect of population density. Okay? The drug acts very differently depending on population density. Now I'll note that all these experiments are done in exponential phase growth. So if you just let the cells grow, they'll get an exponential curve. Or alternatively, if you measure the growth rate as a function of density with no drug there, you don't see any effect. Okay? So a typical sort of textbook definition of bacterial physiology would say, we expect basically the same behavior over this whole range of exponential growth. This is saying that's not at all the case. So we did this for a lot of common drugs, and we studied a lot of different effects. I'll just point out 2 of them. This is a case where the drug fails at high density. So as density increases, the growth of the population increases. The different colors are just different concentrations of drugs. Okay. There are also these interesting cases that look like this. In this case, as cell density goes up, the effect of the drug also goes up. If you have a lot of cells there, the drug actually works better. Okay. In this particular case, we know why it happens. There are other cases that we don't know why it happens. You know, one question that we're very interested in is why does something like this happen? And the answer is, of course, as often in biology, it's complicated. It depends on the drug, it depends on the strain. So there's definitely examples in the literature of molecular mechanisms that are consistent with the drugs we used and maybe playing a role, although they haven't been explored specifically in our species. One of them is drug degradation that I talked about earlier. Another is something related to heat shock and growth bistability. There's also some type of drug sequestering effect that can take place. We actually found a new mechanism, not a particularly surprising one, and given time constraints, I'm not even going to talk about it, but basically what can happen is the community of cells can modulate the local pH of the environment. That local pH change can change the activity of the drug. And so that's one new, although not particularly surprising, I would say, community effect. I'm not going to talk about that specifically. But what I am going to say is, what about the cases where we don't know anything about mechanism? Can we still make any type of important predictions? And I would argue that we can. So think about what's going on here. We have some drug in a vial, and let's say we want to figure out what the drug concentration is in that vial. So we have some input vial that's at a concentration Dn, flowing drug in. We then have drug and cells flowing out to keep the density constant. So we can write down just a continuity equation for the drug concentration in the vial that involves the flow rate and the drug concentration. That's just a continuity equation for conservation of drug. Then we can add to this our density-dependent effects. The way we add this to the mathematical model is say that there's some density-dependent term that decays the drug, or that makes the drug concentration either decrease or increase depending on the effect. And so the point is, can we get at this parameter? And we can. So this is just a phenomenological parameter, but it's really powerful and can tell us something about how treatments might work. Okay, so we take an equation like this. You have this flow rate divided by volume, which is actually just the growth rate. You have to set the flow rate so that it equals the growth rate, otherwise you won't keep the population constant. When you do that and you solve for the steady state, you get an equation. The equation is not completely determined yet because it also depends on drug concentration, which is itself related to growth, which we're measuring, so you have to do a little more work. But effectively what you end up with is an equation that relates our experimental inputs, which are the drug concentration in the input chamber and the cell density, to the output, which is the growth rate that we measure. And to get this relationship, we have one unknown parameter, this epsilon parameter, which we can estimate with things like Marisa uses. Don't tell me this isn't identifiable. And then you can start asking questions about, okay, so maybe we don't know the mechanism for sure, but we know something about the time scale of the effect and the magnitude of effect, and it's contained in this phenomenological parameter. We can now say, let's say we have a growing population. Do we see an effect? If we add just a constant amount of drug, if this were a process that were happening very slowly, the cell population might grow all the way up to some steady state value without anything in the environment effectively changing, right? So we can start to answer questions like, does this really matter for a growing population? The answer, let me look at time again. The short answer is that yes, it does. And the idea is that, you know, we take whatever mathematical model we have of interest, we now incorporate this density dependence, which is something that we measured, and then we can ask questions like, let's say we start at some fixed drug concentration, and we start at some really low population density, and we ask how long does it take the population to reach some threshold value? Right? This could be something that's important clinically under particular conditions. And does the density dependence affect that? And what you find, and we can test this experimentally, is that in fact it does. So this black curve is the prediction from a model as a function of drug concentration, just the relative time it takes a population to reach threshold. So it's not surprising that that increases. Now the exact way that it increases is predicted by the model. The dark spots are just experiments. What we can also do, though, is because this is one of the drugs where we know the mechanism, it's pH driven, and we can turn that mechanism off, we can then ask the same question, except in the case where there is no density dependence. And when you do that, you see that this effect changes quite a bit. That's the red curve. So this is pretty cool. It says the density effect is quite big. You can now take this to more realistic models, like clinical dosing models, where you have some periodic dose of drug over time. And you ask yourself, if you have a density-dependent effect like this, would this really matter for a clinical treatment? So we took these models from the literature, just sort of classic PK/PD pharmacology models. We incorporated our additional density dependence through this decay parameter. And then we asked, what's the long time behavior? Can you clear the infection with a treatment effect that would typically work if you didn't know about this density effect? And while you can easily solve this numerically, we also developed some really simple approximations where we can solve a lot of this problem analytically. So that's kind of nice. And what you get is a diagram that looks like this. On the x-axis is the antibiotic concentration. On the y-axis is the density of the bacterial infection. The blue region here is region of this space where the infection is cleared, and the white region is the region where the infection is not cleared. The red lines are just analytical approximations. And so what you see here is for very low drug, of course your treatment fails. That's not surprising. Same thing happens for very high drug concentration, or it's not surprising in the same way, it always works. But there's this region in the middle where the dynamics are bistable. And what that means is that if your density of infection is sufficiently low, you can clear it. This is population density over time. But even at the exact same pharmacological parameters, if the density reaches some critical density, you no longer can clear it. When you put in numbers here, the effects are potentially huge. There are some cases where it can be 100 times or more the MIC of the drug once you reach a realistic clinical density. So what this is saying is you have effective drug resistance in some cases, at least possibly, at least clinically resistant in terms of a pharmacology model, even without any genetic resistance there, purely from these types of density effects. So this is a paper that just came out where we see a lot more details about this. Came out about, I don't know, a week or two ago. Some of the questions we're looking at in the future is, does this affect the evolution of resistance? So if the density changes how strongly the drug acts, that means it also changes the selection pressure for resistance to pop up. So we're starting to look at things like, how does the evolution of resistance change? For example, not just as a function of density, but as a function of density fluctuations over space. And so I'm not going to go into this because of time, but I have a student doing some of the theoretical work behind this. Briefly, I'll say that it depends on the spatial layouts of density. You can both dramatically speed or dramatically slow evolution. At least that's what these types of models predict. And this is something that what's kind of cool is we can actually measure this. We can set up different density chambers that are all connected together. And if you watch in one density chamber, you can measure the relative growth rate over time. And this is one experiment with ciprofloxacin, I think. In about 10 hours, what you see is a mutant takes over. So the growth is kind of slow. The drug is affecting it. But all of a sudden, the mutant takes over and it reaches not quite the wild type growth, but some faster growth rate. So we think we can measure this in real time, which is kind of exciting. So the hope is that more on this is coming soon. The theory, very strongly makes predictions that we're trying to test. So, I will just mention, for the sake of completeness, some other things we've got going on in the lab is we're trying to understand the spatial dynamics of bacterial biofilms and how things like cooperativity would affect the spatial arrangements of cells in a biofilm. So for example, if you consider this enzyme-mediated resistance case and you take a biofilm of mixed species resistant through this enzyme and sensitive cells, first of all, you'll find that if you apply a drug, you will find that the biofilm has both cell types and survives even at very high drug concentrations where the sensitive cells alone cannot survive. And then if you zoom in and look near those sensitive cells, you see that there are neighborhoods that are statistically enriched for resistant cells, even relative to the global population. We've developed some simple computational algorithms to take data like this, find out the positions and sizes of all the cells, and then tell us what these spatial neighborhoods look like. We also do some things with drug cycling in something called collateral sensitivity, where the resistance to one drug gives you an increased sensitivity to another drug. And then we do some statistical physics stuff. I'll end just by saying thanks. This was work, again, led by Jason, also with Jeff and Peter. And then, funding and things like that. Thanks. Mm-hmm. Yes. That's right. A little bit. So I probably mentioned before, my brother's a cancer biologist, and he said the exact same thing. So he's mostly does in vitro stuff and some animal stuff. So we haven't necessarily thought about it in terms of clinical data specifically, but we're doing some in vitro tests of that type of thing. So just from If you can take the, I see, if you can take, right, or some way you could pull out, you could, okay, right, cool, yeah.
Apart from the population size, do you find that the growth phase that the cell comes from will also affect drug resistance?
Yes, so the question is, does the growth phase, either exponential phase, stationary phase, or this sort of lag phase at the beginning affect resistance? Yes, tremendously. And that's pretty well established. And so if we let cells grow to, say, stationary phase and apply a drug, usually the drug doesn't work nearly as well. So the question we were interested in is what happens during exponential phase. And what we saw that was somewhat surprising was that it wasn't sort of a rapid cutoff when you get to stationary phase, although there may be much stronger effects there, but that there's strong effects as you go. That's also true. It absolutely will depend on the mechanism of drug.
For both of you, about identifiability, what would be a formal metrics that people can use? You know, in journals, there is p-value for significance, and then they sometimes ask reviewer for commenting on the robustness of a model. But short of analyzing data ourselves, we don't know. Sometimes that's a suspicion, but we don't have a metrics.
The Fisher matrix, probably. I mean, that's yeah. But you can probably answer that.
I'm like, I think you can probably all hear me, but I guess we're recording, so I should try and, yeah. But if I understand your question right, you're saying like for things like, you know, statistical significance, we have p equals 0.05 or whatever and blah, blah, blah. Like what about for identifiability? Is there some metric that we can use to say, yes, this is safe to do parameter estimation with, essentially, right? Like, and so, yeah, so like there's you can use like full rank of the Fisher information matrix is a common one, like, but in general, like the structural identifiability question is actually kind of yes, no, right? So like that one you can actually check if you're gonna do the analysis. But then you can also like, the trickier part, I think, in terms of coming up with a metric is that the practical identifiability question is actually kind of gradiated, right? And so like, there you have to decide, you know, your 95% confidence intervals, or you are within X or whatever, you know, like you have to sort of decide, much like with statistical significance, what level is good enough for your area. I mean, I don't know. Yeah. So like you'll have, it is an interesting question though about like if there's a cutoff, like, or a metric that you could sort of use. I mean, you can do things like with the profile likelihood, right, where you can say like, here's the shape of our combinations, and this, if you draw sort of the contour line of what a 95% confidence interval on your parameters would be, you can get that kind of thing and then decide if that's too wide or not, you know. But it sort of depends on your application, right? So yeah, it's a bit unfortunate in that way that there's not a more like it is or it isn't kind of thing. It's sort of, yeah.
I would just send it to you. You can report back.
Yeah.
Have you like ever tried or think of using genetic data in the biostable switching modeling part that you like Dr. Wood talked about?
Yeah, like doing the same kind of analyses but with genetic data and things
like
that?
Yep, because I feel like the biostable switches data can be modeled pretty well if you incorporate some of the genetic data in the drug resistance.
Sure, that's entirely possible. So we're doing things like, you know, we've done some transcriptional analysis, like full genome transcription at different densities. We're thinking about things like this. We haven't gotten very far with it yet because we've just started, but yeah, absolutely. I mean, I think the ultimate goal would be to tie this to molecular mechanism and actually explain how the molecular mechanism leads to the global behavior. You know, when we don't have that, we say, can we still do something with it?
Thank you.
We still have time. I'm just curious, like, what everybody's backgrounds are. How many of you guys would label yourself an experimentalist? Maybe let's ask that question. Or like you do stuff in a lab, you know? How many would call yourselves theoreticians primarily? No, different things. Something else. I'm missing the key categorizations because most people didn't. Huh? Okay, yeah. So Yeah, so like, would most of you call yourselves sort of computationally people? Is that what we would? Yes. Okay. Okay. So
There's the
one.
phrase.
That's the one. Yeah. 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 Okay. Cool. Yeah. And what about scale of thing that you work on? How many people are working on cells? Like tissuey things?
Probably everybody.
Probably everybody. What about like whole beings? Whatever. Yeah. Humans, etc. Populations. I don't know. Yeah. Okay. Huh. Oh, right. Yeah, the other direction. Yes, I started too big. Yeah, exactly. Yeah. Okay. I just was curious. Yeah. Cool. All right.
Now that you've been here several years, how would you describe the domain of network biology? Okay, and how big is it on this campus? I mean, I know 4 of you. There used to be 5, but I assume it's much bigger. And how, you just asked this question. How would you define the range, scope, most important questions about network biology?
Okay, this. How long do you have? But yeah, no. I mean, it's huge, right? And there's a huge strength in network biology here, from Mark Newman to all over the place. So and one of the nice things about here in terms of networks and network biology in particular, I guess, being a thing, is that it's quite cross-departmental. So like, that was one of the nice things about the network cluster hire, I think, is that all of us are sort of situated in different home departments. And I think that's a trend for network biology on the campus in general, is that the folks that we talk to are not all in the same places. They're all across the campus, which I think is actually quite nice. In terms of the breadth of the whole field,
there's probably a lot of people that I would say, are getting awfully close to network biology, but don't consider them network biologists, because you're talking about interacting connections that you could describe with some graph structure that's like a network.
Yeah, I mean, almost everything is a network at some point, right? Yeah. If you're working on biology, you're probably working on network biology in some way or another. Yeah, but I don't know. Yeah, I mean, it's quite broad, right? Like, so.
Yeah, I mean, one thing I like about it is there's very theoretical work on the the statistical physics of complex networks, and Mark Newman is one of the founders of that field, all the way to a lot of sort of clinical level and epidemiology level modeling.
Or your own measuring, like you're doing like actual experimental or epidemiological measurements of networks. I mean, I guess, like one commonality between all of, like with Meg and with our stuff, is all of us are looking at interactions, right? Like, so that whether it's of cells or of humans for transmitting disease or for resistance or what have you. I mean, like, so that's I guess the common feature. But yeah. Or in like Meg's case with Daphnia and parasites, right? Yeah, can be whatever. Vika is very obviously.
What is your advice for us about washing our hands? How often? How aggressively? How long?
Wash your hands as much as you can.
And more importantly, can you tell us who hasn't been washing their hands?
So if we're collaborating with folks in engineering who have built these sensors, and then if you happen to be wearing one of our OPPO sensors, then yes, we can tell you whether you've been washing your hands. But yeah, I mean, hand washing is a like hand hygiene, and a lot of these things. I think there will be like maybe more broadly than just thinking about that. There are a lot of like interesting ways that now we can measure social network, and behavior types, like behavioral data that affects social networks and infectious disease and all of these things that we sort of couldn't before. People's reporting of how often they wash their hands is often quite different from how often they actually wash their hands. And so
these are the ones to worry about.
Yeah, right. Yeah. And so so that can be very interesting. Yeah. All right. Is that everything?
So thank you again for the invitation.
Let's thank both our speakers. Absolutely. So as Gil mentioned in the beginning, this is the second in the installment of kind of panel-like discussion or short talks from speakers who are hired. It's called a cluster biological networks cluster hire. So one thing that, at least for me, is that how diverse it is. I think you mentioned it, like it's a very broad view. But even no matter how specific your area of research is, it's still like what every faculty is doing is so diverse, and we can all take home some lessons from it, like how we can, as you mentioned earlier. So I have not much knowledge about the theory about identifiability of parameters and everything, but something that clicks with me is like, okay, if I cannot estimate certain parameters, there is a way to kind of cluster them together and simplify the model, and that's kind of like you can apply that for your own research. So that's, for me, is just understanding and appreciating the diverse research that everyone's doing, and how that's helpful for me in my work, in my doctoral thesis, hopefully. Yeah.
Yeah, it's a great chance to have two speakers focusing on two different scales of network, and very interesting. A lot of time when we talk about biological networks, for like my work is mostly molecular level. So we're just asking like, what level are you on? When you start with cells, and like we skip like half of the population here. So, but on the other hand, we're looking at like network study has been applied to different scales. We're starting from epidemiology, the population scale. We also have Professor Kevin Wood working on the cell population and the drug effect. And we also have people working on introducing a simpler model and try to simplify the interaction between different genes, which can be super complicated. And we also have people combine all these different scales of a network and try to come up with like actor-based models, and is on the different scales and is quite extensible. And to be exposed to such a broad area of research is it is quite interesting.
Then you have collaborations between the people working on the different things.
Yeah,
you have another
definitely.
network. It's all a dream within a dream.
Yeah.
When you work on it.
Thanks again.